Right. Hi, everyone. So I'll be talking about the common sharp-tailed snake and our project in the San Juan Island National Historical Park, experimenting with looking at patterns of the sharp-tailed snake occupancy. And then at the end, I'll talk about some of the future directions that we're looking at carrying our research. Next slide, please. So the sharp-tailed snake, just to catch everybody up, if not everybody's familiar with it, it's a very small semi-fossorial snake. You can see these two pictures here, the one on the left and the one on the right, showing the dorsal and ventral colorations, as well as particularly the picture on the left. You can see the nominative sharp tail there. It's the only snake in Washington that has a tail spine. But these are small snakes. Their distribution ranges from BC down through California, but the more northerly you get in the latitudes through that distribution range, the more spotty the actual populations begin to get, and the less data we have from those populations. So in Washington, there's less than 24 known distinct populations in the state. And in Western Washington, they're pretty rare. Several populations are known from the San Juan Islands, and there was one population described in Pierce County. But one of the habitat types that, at least in Washington, the snake it tends to be highly associated with is Gary Oak savannas. So that's that picture in the middle there. And if we go to the next slide, so we see this is a map here that was current as of 2019, looking at the distribution of sharp-tailed snakes in the San Juan Islands within the last several months. This needs some updating, actually. Stewart Island got its first observation from the San Juan Islands Preservation Trust a few months ago. But if you look at the table here, you'll see that the San Juan Island National Historical Park, the Young Hill unit, had a few observations made in 2016. That was part of an effort to, after initial observations were made in 06, 07, some subsequent efforts were made using cover objects and cover object surveys on Orcas Island and on San Juan Island to sort of get a sense of what the distribution of sharp-tailed snakes was in the islands. So Young Hill had 3 observations found from cover object surveys in 2016. So if you go to the next slide. So Young Hill is characterized but it's a sort of a mixed habitat, but it has a large south-facing sort of rocky, grassy oak prairie that you can see in these pictures here. And that's one of the habitat types that sharp-tailed snakes are associated with, typically found sort of around the edges in particular of these types of habitats. And around the margin of this open grassy slope on Young Hill is a mixed forest comprised of Douglas fir, Gary oaks, big leaf maples, and in some places arbutus, the madronas. So you can see in these pictures, the one on the right hand side, that's a drift fence array. So that's sort of where we had our experimental cover boards that we put out, which I'll talk about with the next slide. So in 2018, we did some opportunistic surveys of old cover boards, those original cover boards that were put out. Those artificial cover objects were asphalt roofing tiles. We didn't have any observations of sharp-tailed snakes during that year. So in 2019, we deployed new ACOs, or artificial cover objects. So rather than using the roofing tiles, which had some success, but it was somewhat limited. We employed 2-foot by 4-foot boards of untreated plywood. That's 1/2-inch CDX 4-ply boards. And we used a mixed treatment within the Gary Oak habitat, having 5 boards out in the open, and we had 5 placed within the forest edge under mixed canopy cover. And from 2019, when they were deployed through 2023, we had weekly surveys of those cover objects. So next slide, please. So here's a quick summary table of our observations. So in 2019, we had 1 observation that initial field season. And then the subsequent 2, we had 12 observations in 2020, 17 in 21, 7 in 2022, and then 26 observations this past year in 2023. So you can see I've marked that first one in 2019. The boards weren't deployed until June. So a significant portion of the spring sampling window didn't happen because of that deployment date. And then in 2022, I've got that marked out there because there were limited spring surveys due to some logistical issues with getting people out in the field that spring. But if you look at the picture here, the satellite image, you can see the actual placement of the boards within the habitat. The left-hand rectangle encompasses the 5 boards that were placed within the forest edge with that canopy cover. And then on the right-hand side of that, that second polygon has the 5 boards that are out in the open, sort of set out from the forest margin. So next slide, please. So we had 63 total observations from 2019 to through the end of 2023. These are what they look like if you break it down by month. So that spring period, sort of March through June, tends to be where the vast majority of our observations are clustered. And with fewer after that, July through October. And the significant thing here is this lines up pretty well with the conditions moving from spring to summer, moving from sort of a wet weather regime to a drier one, the surface soil becoming drier, less humid, and temperatures going from cool to warm to hot there during the summer. So we see that sort of lines up with a decreased observation frequency for the sharp-tailed snakes. So if we go to the next slide. So here's a heat map of those 63 observations that we had. You can see the ones clustered in the open on the right-hand side. There were 44 of those 63 occurred out there. And then we had 19 that occurred inside the canopy cover. So a statistically significant difference between where the snakes were choosing to appear under those cover objects, with quite a bit more selecting that more open habitat that's sort of comprised mostly of grasses. There's some trailing blackberry, Oregon grape, some small shrubs like that, but mostly grasses. So next slide, please. So if we look at that first period, that earlier portion of the year, while it's still somewhat wet, we see a significant number, significantly higher number of the snakes appearing in those open, in that open cover board treatment versus the cover boards that are within the canopy cover, within the forest edge there. So there were 55 out of the 63 were found here during this time period. And then if we go to the next slide, that later in the year period, we had a total only of 8 observations, and they were evenly distributed between the cover boards placed in the forest edge and then the cover boards that were placed out in the open there. So next slide, please. The future directions of where we'd like to go with this work would be to place some additional cover object arrays on the Young Hill unit on the national park, and also on Orcas Island. We've installed some cover object arrays to get a sense of how different covariates on different islands in different specific habitats might be driving occupancy patterns. We have deployed temperature and relative humidity sensors for the surface conditions underneath some of those cover objects. So we can hopefully look at specific drivers beyond just open canopy cover and closed canopy cover treatments. And one of the things that we would hope to build in is population level analysis, including survivorship, which would be predicated on using robust photographs of the individual snakes that we capture, using pigmentation differences and permanent sort of marks like stub tails or scarification to identify individuals within seasons and then between seasons to conduct recapture analysis. But one of the other big things that we'd like to look at is looking at fecal analysis for DNA residues for prey identification. Sharp-tailed snakes are predominantly known to feed on slugs. So being able to, and of course, with the pattern of occupancy that we've seen so far, I think it's likely that the availability of prey, slugs and soft-bodied invertebrates decreases during that latter part of that chart that we saw, the July through October, which is likely tied to the snake occupancy at the surface. So being able to build in some prey analysis into occupancy models would be a direction that we certainly want to go. And then working on genetic analysis to look at look at the relatedness of the populations within the islands, between the islands themselves, and then between the islands and the mainland, where some genetic analysis and individual haplotypes have been identified just using swab samples. And then looking at fine scale movements following recaptures is another target of ours, looking at how their specific individual snakes are moving within the habitat post recapture. So next slide, please. And I'd just like to thank the National Park Service and WDFW, the San Juan Island National Historical Park, and then all the QIAT staff and volunteers and interns that have helped out with this project.